Good morning, everyone. For today's agenda, we will have poster competitions and we will have Sir Edwin from Biology ITS for our judge. He has a doctorate degree in geobiology in Ludwig Maximilian University Munich, and he is an expert in sponge research. Sir Edwin, hello. How are you?
Yeah, not so good. I got some sore throat and fever, so it's okay. Yeah, luckily we conduct it online.
Yeah. Okay.
Yeah,
Sorry. I hope you getting better soon.
thank you.
Okay, sir, shall we go to our first presentation?
Yes, sure. So the first group, go ahead to your presentation then.
Yeah, okay. You guys have time for the presentations about 15 minutes, and for the Q&A sessions from Sir Edwin, we will have about 10 minutes. Okay, group one, are you guys already complete, all the members?
Not yet.
Group
No
one?
Excuse me.
respond.
Excuse me.
Yeah.
Sorry, ma'am. Our group is not complete yet. We
Okay,
still
number
contact the
two
other
then.
member. So
Number
sorry.
two.
How much who hasn't been in from your group? Nathan? How many person who
Excuse
hasn't
me. Is
in?
already complete now.
Okay, okay, you guys can start the presentation.
Excuse me. I want to share my poster. I'm sorry. I have some problem here. Maybe is my friend can do a share screen.
Is there a problem from group one?
Okay.
Okay.
Okay. Is it already seen?
Yeah,
Is
go ahead.
Okay. Thank you. Okay, first of all, thank you for this opportunity. My name is Hari Wijaya. I'm from Group One. With my friend here guys. Is it seen clearly or is it some blurry? Miss Tina, can you help me to?
Nathan, I'm sorry, I didn't catch the words you were saying. Maybe you can speak louder.
Okay, is my screen is clearly or it's some blurry?
Okay, it's clear. It's clear.
Okay, thank you.
Okay.
I'm from Group One. My name is Nathan Wijaya, and my friend here is Jason from TAU, not from TAU, and Singrati Firdaus from ITS. In here, we have topic about enzymatic modification of biomass, and the topic that we raise is about enhancing enzyme mediated cellulose hydrolysis by incorporating S group onto the lignin during biomass pretreatment. Maybe from the first part, maybe from Matt will explain about the introduction. Okay, Matt, the time is yours.
Thank you, Nathaniel. Am I audible, ma'am and sir?
Yes, you are.
So, I will discuss in the part of the introduction. So we have here in the figure one the various lignocellulosic agricultural waste and crops. So first we have here the wood pellet, wood chips, rice straw, wheat straw, palm waste, sugar cane, bio-wastes, and short rotation coffees. So here we have a 200 million tons biomass waste almost generated in many countries in 1 year, according to Singh et al. In 2014. So a huge biomass waste of almost 100 million tons is generated in many developed and developing countries in 1 year. So taking the example of one of the most populous countries like India, they have a large metropolises generate 6,000 tons of solid waste per day. So India itself generates a 657 million metric tons agricultural biomass per year, whereas the forestry and wastelands residues are 260 million metric tons per year. So in the second figure, we have here the wood chips, specifically the aspen woods that is used in this study. So it has a wood composition of 69% of oleocellulose and 18.6% of pantothans. We have here the 17% of clays and lignin and 1.2% of ash, according to Bollock et al. In 2008. So in the next figure, the figure 3. So this is the schematic representation of legume cellulosic biomass pre-treatment. So here, the pre-treatment of legume cellulose by different methods removes the hemicellulose and lignin from this matrix of polymers in which cellulose exists before hydrolysis. So lignin has been proved to be a significant obstacle when trying to carry out effective enzyme-mediated hydrolysis of lignocellulosic substrate. So lignin binding to the cellulotic enzymes and limiting their activities according to Wu et al. In 2022. Then we move to another arrow. So we have here the oxidation and sulfonation. So this pre-treatment method that's add more acid groups and aliphatic hydroxyl groups, but less phenolic hydroxyl groups. So these two methods of both approaches are widely utilized by the pulp and paper sector because oxidation and sulfonation are the four most common methods used as this modifies the lignin structure and charged by the incorporation of acid groups. Then lastly, we have the enhanced lignin hydrophilicity and increase the negative charge, decreasing the non-productive binding of cellulases to lignin according to Wu et al. Also in 2020. So for the research purpose, Natalie, can you move upward for the research purpose? So for the research purpose, as you can see, it has to determine the effectivity of acid groups by incorporating onto the lignin during biomass pretreatment in enhancing mediated cellulose hydrolysis. So that's all for me. Thank you.
Okay, so in the next, I will explain about material and methods. So that material in here is there are several materials. There are biology material is aspen wood chips and cellulose related liquid craft pulp that we use from this method and in this study, and the enzyme that we need is protease enzymes and some chemical like sodium bicarbonate, sodium sulfate, and anhydrous ether. So in this study, to to uh to do some enzyme modification, we do some a pre-treatment. The first pre-treatment is water-based heat. In here, 20, 250 grams of aspen chips were soaked with water in the cooking vessel at a liquid the fat wood ratio of 4:1. The cooking vessel must then move into the digester. The The temperature was hit to 170 degrees of Celsius within 1.5 hours and stay at 170 degrees of Celsius for another hour. The vessel was then pulled down in the cold water. After we do this water-based heat, we move to the acid wash pretreatment. In this pretreatment, the hot acid pretreatment was performed using the method of Furinon to minimize the loss of viscosity and physical strength. The pulp samples about Aspen was ships before that already was due by water-based heat is treated with a sulfuric acid solution in pH 3.0 at 95 degrees of Celsius and for 3 hours. And after this pretreatment, the pulp was washed thoroughly with deionized water. So after this pretreatment, we do the first treatment with alkaline oxidation and neutral sulfonation of pretreated aspen. So 20 grams of pretreated and acid was mechanical pulp that we have to do before was incubated overnight with either 16% sodium sulfite or 32% sodium bicarbonate at 10% solid loading in a 60 degree of Celsius water bed. The substrates were washed with water after the reaction and stored at 4 degrees of Celsius for further analysis. After this oxidation and sulfonation that the oxidation and sulfonation is the aim of this study. So we need to analyze is it really affect about the enzyme hydrolase modification. So in the next step is enzymatic hydrolysis. In here, the substrate at 2% solids is loading in 2 milliliter of cup centrifuge tubs containing sodium acetate buffer and the salicity AC3, and then the tubs were incubated in a rotating incubator at 50 degrees of Celsius, and the released sugars measured using a glucose analyzer. After 6, 24, and active a 48-hour of
Nathan,
hydrolysis.
I cannot see your presentation. It's blank now. So nothing to see. Yeah, tidak. Okay.
Sorry, is it already seen now?
Yes. Yeah.
Okay, I'm sorry. Maybe my connection is bad right here. Okay, so after that, we do some substrate characterization after the enzymatic hydrolysis. In here, the chemical composition of the substrate was analyzed according to the class 1 protocol using PAPPI standard method. After that, we do some protease split lignin or PTL isolation. The split-treated substrates were subject to enzymatic hydrolysis before is using the salicity AC3 preparation at 2% solids and protein loading of 30 milligram per gram cellulose at 50 degrees of Celsius for 72 hours. The hydrolysis residue was centrifuged, was and subject to a second round using enzymatic hydrolysis in the same condition. After that, the residue was incubated with some protease in phosphate buffer at 37 degrees Celsius for 24 hours. After that, the protease with lignin was passed through a 40 mass screen and freeze dried. After that, we use some enzymatic metacidolysis lignin or EMAL isolation. This is extract from the protease treated lignin samples that we have flash-treated before. So the mixture was then centrifuged at the and the supernatant collect and precipitate anhydrous ether, some chemical compounds that we used before. After that, the lignin has settled in. It was filtered through a polyphenylidene fluoride membrane. We use some membrane was with anhydrous ether and dry in 40 degrees Celsius of vacuum oven. After that, we do some tests for analyze the substrate. We do some hydrophobicity test of lignin and zeta potential measurement of lignin. Maybe we can skip this method because it's only a test, and my friend will explain about the result of the test. After that, we choose some absorption of lignin to advanced enzyme cocktail using salicitac 3. In here, the sample was performed at a 10 degree of Celsius in 2 milliliter cube cup centrifuge cups using 1 milliliter of sodium acetate buffer. After that, the vials containing 1% lignin and enzymes were incubated for 3 hours, followed by centrifugation and the supernatant will collected. After that, the amount of enzyme absorbed into the lignin was determined as the difference between the initial enzyme loading and the free enzyme present in the supernatant. And the last method that we use in here is gel permeation chromatography or GPC. So in here, the molecular weights of E and ER that we have to before is analyzed by GPC using an epsilon 100 and 1100 cube with an optilat. After that, the data were collected and analyzed. So maybe this is from the methods. We can move to the result and discussion.
Okay, so for the results, is the first result is about the influence of lignin sulfonations and oxidations on cellulose accessibility and enzymatic hydrolysis. Here, as we know that the presence of hemicellulose and the inherent acid groups associated with the biomass might also influence substrate swelling and cellulose hydrolysis. The pre-treated aspen slurry was subsequently pulped using a twin-gear juicer to simulate mechanical pulping. As initial conductometric titrations had indicated that the pretreated mechanical pulp still contained some weak acids, the pulp was subsequently acid washed to remove any residual uranic acid groups that were potentially located on the residual hemicellulose. The subsequent alkaline oxidations and neutral sulfonation reactions of the acid group free pulp was carried out using sodium bicarbonate and sodium sulfite, respectively, as alkaline oxidations and natural sulfonations had previously been shown to enhance the enzymatic hydrolysis of pre-treated pulps. As we hope to modify the lignin rather than remove it, the reactions was carried out at 60 degrees Celsius to minimize solubilizing the more hydrophilic oxidized or sulfonated lignin. And we can see here in the table 1, in the sulfonations and oxidations treatments, a chemical loading of 16% sodium sulfite and 32% sodium bicarbonate resulted in the incorporations of similar amounts of sulfonic and carboxylic acid groups while recovering more than 90% of the original water insoluble component like what we can see here in the table 1. However, as has been suggested previously, it is likely that the majority of the carboxylic acid groups present in the sulfonated substrates were located on the hemicellulose component, while the carboxylic acid groups associated with the oxidized substrate were associated more with the lignin. As and here we can suggest that hydrolysis could be enhanced at an elevated pH by further deprotonating the acid groups and consequently increasing the lignin's hydrophilicity and negative charge. However, the hydrolysis of both the control and modified substrates was only slightly enhanced when it was carried out at pH 6, like we can see here in the figure 1 beside the table 1, to ensure that activity of the cellulases was not compromised by the use of an elevated pH. A cellulose rich, the linified Kraft pulp was hydrolyzed at both pH 4.8 and pH 6, as we can see here in the figure 2. As observed previously, it was apparent that slightly better hydrolysis was achieved at pH 4.8. And then the next is about the isolations, characterizations, and influence of control and modified lignin on enzymatic hydrolysis result. We can see here that the methods that is used here is using a cellulose cocktail to remove the carbohydrate components to produce a lignin-rich residue, which is subsequently treated with protease to remove the enzymes. Despite the removal of much of the carbohydrate by the enzyme treatment, it should be noted that some residual material was still associated with the lignin, as we can see in table 2 here. When the modified PTL or protease-treated lignin were added to the hydrolysis of the cellulose-rich Kraft pulp with the ratio 1:1 at both pH 4.8 and 6 in the figure 3, it was apparent that the modified PTL were both less inhibitory with better cellulose hydrolysis achieved at pH 6. When the absorption isotherms of the cellulose enzyme is in the presence of the PTLs were measured at pH 4.8 and pH 6. It was apparent that sulfonations and oxidations both decrease the extent of absorption between the enzymes and the modified lignin. And as it was possible that the residual cellulose associated with each of PTL samples might influence lignin analysis and structure, we also used the enzymatic mild acidolysis lignin or EMAL method to isolate lignin at relatively high yields without changing its structure and properties. Subsequent gel permeation chromatography or GPC analysis of the EMAL samples indicated that both sulfonations and oxidations resulted in a slight increase in the molecular weight of the lignin components, suggesting that the lignin underwent mild repolymerization reactions under alkali conditions. Here we can see in the table 3 and in figure 4. Interestingly, both sulfonations and oxidations resulted in a slight increase in the aliphatic hydroxyl group content of the lignin, which was probably due to the cleavage of the ester groups connecting the lignin aliphatic chain and carbohydrates during the sulfonations and oxidations of lignin. The observed increase in carboxylic acid groups attached to the EMEL lignin further confirmed that lignin oxidations and carboxylic acid groups had attached to the email lignin and then had occurred in the table 3. So this was consistent that with previous studies which had suggested that the aliphatic hydroxyl groups in the lignin made it more hydrophilic, thereby reducing enzyme binding. In contrast, a greater amount of penophilic hydroxyl group appeared to enhance enzyme binding.
Hello, hello, am I audible?
Yes, you are.
Um, in conclusion, both enzymes accessibility and cellulose hydrolysis may be enhanced by the addition of and, and acid groups onto the ligand present in biomass substrates. It absolutely was apparent that both oxidation and sulfonation decreases the extent of the enzymes binding with the reducing within the number of phenolic hydrolysis groups in similar balance of incorporation of acid groups enhancing ligand hydrophilicity. Consequently, enhancing cellulose hydrolysis. Uh, is the last of the acknowledgement. First and foremost, praises the thanks God Almighty for His shower of blessings. We thank for the support of Institute of Technology, Sepul November, and Tarlac Agricultural University. To the undertaking could not having possible within the participation of teamwork. We would also like to thank the Museums, Davidson CA for providing the enzyme used in these studies. And here's our the reference. Thank you.
Okay, thank you, Group 1, for the presentations. Now we will have Q&A session from Sir Edwin. Sir, you may give the questions to the Group 1.
Yes, thank you, Dinar. I would like to ask first, why you use the aspen wood chips? Is there any wood or other woods? Because as far as I know, if you use wood, all of the wood or plant has the cellulose. Why you choose the aspen wood chip here? Yeah, why you choose the aspen wood chips? Why not the other wood, for example?
Okay, thank you for this. We can uh, answer the question, or
Hmm?
we need. Okay, Sir Edwin, I will try to answer your question. Thank you before. Also here in this study, we use aspen wood chips because aspen wood is one of serious biomass that is produced this um, in this city of the outer. So the aspen wood chip is consists of many of cellulose and polycellulose that contain 69%, and calcium lignin is 17% here. So because the aim of the study is enhancing enzyme-mediated cellulose hydrolysis by incorporating acid group onto the lignin, so we need to use some product that really much of cellulose and lignin here. One of the product that the author use is a wood chips that is already the author is live in Canada, and the Canada is
Hmm.
the serious biomass waste in there is these wood chips rather than a new crop biomass. So they use this aspen wood chip because it's um, there is more than any other biomass. Maybe like that. My friend maybe will add some quick answer maybe.
Second question, if there is no, okay, I would like to ask why here. What is the zeta potential? I don't understand. What is the zeta potential? What is zeta potential on your table? In the table there is in the result there is a zeta potential. Is it measurement for the lignin or something like that, or I don't know what is zeta potential.
Okay, thank you, Mr. Alvin, because it's in my part. The zeta potential is this is some a method to um, measure some of the suspension, and here we use because we want to research about this lignin, the lignin composition with we have already used some pretreatment and any other treatment here. We want to test um, the lignin already the lignin residue that already decrease in this substrate. So the zeta potential is a measure how much is of this suspension is um, result. So maybe like that. In here we can see this zeta potential. This is in pH 4.8 and pH 6, and the most effective here is the in pH 6 like here, because and about another treatment, the cellulose hydrolysis in pH 4.8 is not so good, and this uh, well in pH 6 is more high like this.
Okay.
The enzyme activity, I mean, maybe like that. So admin.
So as a conclusion now, what is the important point? So you want to use the what kind of enzyme for hydrolysis? What kind of enzyme? Is it sulfonated, oxidized? Because you wrote enzymatic hydrolysis of cellulose rich. As far as I know, you wrote binding strand. No, not this one. Enzymatic hydrolysis cellulose rich. Where you put 2%. What is 2% there? Just go down. Go down. Yeah, that's you wrote 2% solids. What is that? What is 2% solids? It's not quite clear. Nobody answer? No understand? No?
Group 1.
One, okay. No, it's okay, no problem.
Excuse me, sorry, Mr. Edwin. Maybe we need to we need to some discussion for answer the question.
Yeah.
Maybe you want to ask another question while we discuss this.
No, it's okay already. 3 questions and already 10:30. I think it's enough. Yeah, 20 minutes. Yeah, dinner. Yeah.
Okay, so we are going to have another group to presentate their poster. Thank you, Group 1, for the presentations. Now, for Group 2, you may presentate your poster.
Fresh water.
Good morning, Mr. Andy, Mr. Edwin, and Miss Dinar. I'm Stephanie from Group 2, and here my friend Rico Obligado, Aaron Rex, Erickson, Akil, and today I'd like to present our poster, and this is our study or research from the journal about breakwater agriculture site selection, Maharashtra, India, using the technique of remote sensing and geographical information system.
Here are the introduction. So, aquaculture has become the world's largest growing food industry with an annual growth with the annual growth of 10% compared to 2 to 3% of other major food sectors. So the first step for scientific and sustainable development of brackish water culture is the better site selection, followed by improved culture management. So a number of aquaculture practices are used worldwide in 3 types of environment. We have the fish water, brackish water, and marine. For the great variety of culture organisms, freshwater aquaculture is carried out either in fish pond, fish pens, fish cages, or on limited scale in rice paddles. Brackish water aquaculture is done mainly in fish ponds located in coastal area, and the marine culture employs either fish cages
or sustain or substrate for mollusks and seaweeds such as steaks, groups, and raft. Uh, s- Culture system range from extensive to intensive depending on the stock density of the culture organisms. So the level of inputs and the degree of management in the countries, which in India, where the government priority is directed toward increased fish production from aquaculture to develop to meet the domestic demands, either as a result of a lack of access to large water bodies or over aquaculture practices are also uh, exclusively oriented towards production for domestic consumption. Um, Presently, only 13% of the total brackish water area is equivalent of 1.19 MHA is under aquaculture. Salam et al. 2003. So the remote sensing is the science and art of collecting data on brackish water by technical means on an object provided useful information, which is the Geographical Information System or the GIS. So the application of GIS, remote sensing, and GPS techniques can develop a multi-criteria decision-making system for site selection of brackish water, aquaculture of Pineus monodus. Next.
Okay, and the next is the method that study set from this journalist from that take place from Baika Taluk, then district of Maharashtra, India. That place the researchers because this place have a good potential of aquaculture that we know from their geographic. So and the next is the material that what identificate that from from a computer based map data was collected through field-set imaginary that have any parameter. There are first good data when the image takes the data, it can display good and clear condition on six main parameters. There are engineering parameters, cell quality parameters, water quality parameters, infrastructure facilities, meteorological parameters, and social conditions. And the next parameter is assessment and reasoning. Parameter assessment and good reasoning of data from using two categories of limits, the water and soil quality parameters. And then by using choice analysis to create a composite map and finally to create a final sustainability map. So and in the preset, the research flow chart. So the material used to the research flow that is have a 6 point of step. That research flow chart there have a cover map was extracted from the list tree, and the first step is procurement. That procurement of ARS list tree files, color composite paper print, and then extraction of land use or land cover map using handheld GPS, and then location site which are accessible and have aquaculture potential within a distance of 3 kilometers from the water source. And the next location site, which are split. And the next is collection of data, like with primary and secondary from the different sites, and then completion of data from the different sites for this analysis. The last allotment of weight for the different parameters. From compilation of data for this analysis make make it that have uh, uh, that to creation of the thematic maps, and then uh, integration of all the thematic layers, thematic maps that must to regrouping of 8 layer, and the last to make finally suitability map or a priority map. So, and the next part that will present by my friend.
Okay, for the results, this study revealed that of the study of 20,431.034 hectare is identified as highly suitable with 0.377%, and suitable with 9.873%, and moderately suitable with 1.772%, and unsuitable with 85.027%, and already under aqua farms with 2 point, sorry, with 2.951%, for overall with 100% for the overall total of study area. And from the materials that we use, in figure 1 we can now use we can see now the I can sorry, the figure 1 is land use classification of the study area, and we can now see here the legends such as agriculture areas and others. And in the figure 2 is the classification of mud floods. Next. Sorry, Steph, can we see the figure 6? Yeah, that's right. Through using the materials that were used in the study, we can now identify the unsuitable, moderately unsuitable, suitable, highly suitable, and the existing aquifers in the study of it. And I will give the time for my friend to explain further.
Um, Analysis of data on engineering parameters, soil quality, water quality, infrastructure facilities, meteorological data, and social restriction with the help of GIS techniques resulted in a map showing the suitable areas for the improved method of shrimp farming. In this study area, Lands adjustment to mudfloods are used for agriculture by making partition deck to prevent the entry of tidal water. Um, Government has also leased out some of those lands for aquaculture purposes, so no buffer zones were created between mudfloods and agriculture area. As the land falls under the coastal zone, the study is mainly suggested for the improved method of shrimp farming, as the accuracy of the results of this kind of the studies is directly dependent on the quality of input data. Um, Updated and accurate thematic information are mostly desired. In this study, the parameters were considered mainly for Pineus monodon species, but for the other species, the same parameters can be used with some modification. Um, Data on water quality parameters are needed at least for 1 year along with meteorological data for the same year so that it would be possible to correlate the water quality data with meteorological data. Um, this will help in assessing the changes in water quality, especially in salinity from available meteorological data, particularly rainfall data. It is possible to produce shrimp across the wide concentration ranges of soil properties. Um, this study has identified 2,456.145 hectare, 12% of total land area of the study region in Palgar Taluk of Tain district, suitable for shrimp farming. From the identified 2,456.145 hectare area, um, 76.95 hectare is highly suitable, and 2,017.13 hectare is, sorry, 76.95 hectare is highly suitable, and 2017.13 hectare is suitable, and 362.65 hectare is moderately suitable. The results of this study were confirmed by the fact that many farms already exist in those areas, which were classified as suitable for brackish water aquaculture. At this point, it is up to mention that further development of these areas already having farms must be one with caution, as unregulated proliferation of farms may exacerbate for a risk of pollution. In general, GIS analysis shows that the major constraint to aquaculture are excessive salinity in some areas and social restriction in others. That's all to me. Thank you.
Thank you, Aaron, for the explanation about the result and report. So the conclusion of this study. And the study demonstrated the potential use of remote sensing and also GPS system
Connection is not very good, I think. Yeah, Tina, there is a bad connection.
Yeah, I guess her signal is not that good. Wait. Hello, Stephanie? Or maybe your the other group members can read her section.
Oh, okay. It's just,
So sorry for that connection.
yeah, it's okay. No problem. Yeah.
This is our conclusion The weighted overlay method adopted in GIS to the potential area into four major categories as highly suitable, suitable, moderately suitable, and unsuitable. And important parameters such as accessibility to the site, topography, weather source, tidal amplitude, soil type, and availability to offset marketing, etc., are sufficient to identify the suitable areas for aquaculture of national level like this in India. And however, the number of parameters to be considered will also depend upon the type of farming system and also freshwater or brackish-water aquaculture and extensive or modified extensive or semi-extensive or intensive aquaculture. And that's all from us, and this is for the references. And thank you so much for your attention. And I would like to apologize if there any mistake in our poster presentation. So,
Yeah.
Okay, thank you, Group 2, for the presentation. Now we will have a Q&A session. Sir Edwin, you can go ahead ask the question
Yeah,
for
thank you, Rina.
Group 2.
Okay, now what is the definition of GIS? You know, Geographical Information System. So what is actually that? You use the satellite or you use the mapping, and then could you just explain to me why GIS is important, the basic principle? Because you show me the map, brackish water, fresh water, salt water, and then you give a map on that, which one potential, unpotential, sustainable. So what is the basic principle of GIS?
I want to answer the question. Stephanie from Biology ITS. And to answer what is GIS, data layering is a method that this method have a main principle to organize the data that we have a special data layer. So we have five or four data layer, and data are organized such the similar elements are a single data layer, and we also use fast color composite, and this fast color composite we use to know the vegetation or the layering of the site for the aquaculture, and we can detect easily because the human eye cannot see near infrared wavelengths. So here we use false color composite, and so the use of near infrared wavelength it can improve the spectral separation, and the data can be better to be interpreted. Maybe that's all from. Maybe my friends want to answer.
Yeah. I just want to add on its definition, GIS or Geographic Information System is a computer system for capturing, storing, checking, and displaying data related to position on Earth's surface. So in this study, they use GIS to through the help of satellite images, and then they compile it. And for that, they able to provide the suitable maps for brackish water culture area in the India, so that the farmers or the farm growers they have the guide, they have the guide on where the areas that is possible for brackish water culture in their area. I think that's it.
Yeah, thank you. So yeah, so all of them are based on the satellite and graph, yeah? Yeah.
Yes, sir.
Yeah, okay. Okay, thank you. Now second question is about how you categorize like this one on the study, which one suitable, highly citable, moderately citable. So what is the parameter on deciding that? Is that any maybe because of the geographical or the plantation or what? Why this in this journal? What is the parameter to decide whether this one is suitable or not? Yeah, is that okay? How you distinct that on the suitable or unsuitable? Anybody has read the paper? That's why I don't understand why they put maybe because of the plantation or the surface of the pond or the water. Is that any idea on that?
Okay, Victor, thank you.
Yeah.
Yeah, to answer why we uh,
Yeah. Yeah, Muhammad, go ahead.
why what is the parameter that uh,
Oh,
the parameter derived uh, from the like engineering parameter like topography, uh, drainage facility, water supply, like that, and then from soil qualities, that from like a soil pH, percentage of clay, and then organic carbon, nitrogen, and others. And then from water quality, like this place is a good water temperature, dissolved oxygen, and the next infrastructure facilities that in this area is good facilities from the accessibility, land value, and marketing facilities, data power facilities, and um, the availability of technical guidance. And then from the meteorology, have a good atmosphere temperature, good rainfall, and the last is from social restriction, that restriction from the village and government. So like that.
yeah, thank you. So it's a lot of parameter like village. Okay. I think it's one question, still 2 minutes. Let me check again here. So here, as in the conclusion, we wrote remote sensing, GIS, and GPS. What is the difference among that? GPS, GIS, remote sensing, is it the same? As far as I know, is almost the same, but a different principle. Have you any idea on that? On the conclusion? GIS, GPS, and remote sensing.
Maybe I can answer education.
Yeah.
What is the difference between remote sensing and GIS? Before we use or do the GIS, or we should do a remote sensing. Sensing is one of the methods commonly used to collect the physical data of the place or the site, and the data will be integrated to GIS. And the this remote sensing method, it collects data from an object of the Earth in the site and without direct contact. So the remote sensing use some wavelength. So the remote sensing can interpret the data and the GIS can visualize the data. Maybe that's from.
Okay. Thanks, Stephanie. Clear now. Okay, I think it's enough dinner, yeah. 11 o'clock. So we are already on time. Now the third, yeah, thank you very much for the second group. Thank you.
Okay, thank you, Group 2. And now we're moving on to Group 3. For Group 3, you can start your presentation.
Vaisaka, Dinda. Okay.
Okay, am I audible, ma'am, sir?
Yes, Vaisaka, go ahead.
Okay, thank you, Pak Edwin. Good afternoon, Bapak Edwin, Ibu Nailis, and Mbak Dinar. And also, good afternoon to fellow biology ITS and target agricultural university students in the Philippines. At this time, we are from Group 3. We'll be presenting our poster with the theme Fish Parasitology entitled Identification of Parasites and its Prevalence from Grouper Commodities collected in Buleleng Regency, Bali, Indonesia. Our group consists of Waishaka Gandra, Tinda Shafitri from Biology ITS, and Avijita and Karda Kumos from Agricultural and Biosystems Engineering TIU. Okay, we are going to the introduction section. Maybe from Avijit, are you there?
The first is introduction. Since 2014, the total production of the fisheries sector worldwide has reached 167.2 million tons, which is 44.14% of time from aquaculture. Intensification is an alternative program to increase aquaculture productivity on limit line. Intensive aquaculture system with high stocking of homogeneous fish can trigger the emergence of various diseases, especially parasite. The parasite infection to fish may affect its quality, thus can reduce the number of export for some commodity due to the low quality of the fish product. Gobber fish is one of Indonesia's leading commodity in the field of marine culture. This commodity is sold on the export market and still has a high potential to be developed. In some places, the effort to grow gobber fish problem include tools that occur in Buleleng Regency, Bali. Most of the pests and diseases that have infected gobber cultivation are parasitic infection. The second is research purpose. According to this method, this study I have to monitor the prevalence of parasites contained in the global commodity as cultivated, consumed, and as ornamental fish in Buleleng Regency, Bali, Indonesia. The method, one study area and a counter problem. The study was conducted in a global community in Buleleng, Bali, Indonesia. The problem, the decreasing in total export due to the low quality of the commodities. The second is test material. The type fish used for monitoring was grubber fish. They are fry, consumption fish, and ornamental fish. which were taken as many as 80 fish samples. The research design, the study was conducted with a sample random sampling method. A total of 80 fish samples that are fry, consumption, ornamental fish were used for identification and prevalence of parasite. A discussion was present using a descriptive method, and the last in method is observation of parasite. Necropsy was performed to all collected samples. All samples were measured in length and weight, and external clinical symptoms were observed directly to organism. External organs, scalp, fin, operculum were taken for parasitic examination. Parasitic infection was investigated under a binocular microscope. The type of parasite was morphological identified, and the prevalence of parasite infection were calculated using the formula. The formula, which can be seen on the poster, prevalence of parasite is number of infected fish per number of fish observed multiple 100%.
Okay, maybe Dina can scroll. Okay, thank you. Okay, now I will explain about the results that based on the research, there are three results that I will explain. The first result is talking about the inspection of the group of commodities. Based on the results of examination and identification of parasites in 80 group samples from fry, consumption, and ornamental commodities, there are two types of parasites. These parasites were found in the gills of examined sample. The grouper community that didn't experience parasitic infection was the ornamental grouper community, which showed no parasite from all the three samples. You can see the result of examination of parasite in 80 grouper samples in the left figures. Sorry, can you scroll a bit up? The first one, the first table. Okay. The first, can you scroll up, please? The first table. Okay. We can see in this table there are three commodities, three group of commodities. The first are the seed or the fry commodities, and the consumption commodities, and the ornamental commodities. From the fry or seed commodities, we can see that about 50 fish are the free or non-infected fish, and about 8 or 9 fish are infected with the parasites. And in the consumption fish commodity, there are about 20 fish that are not infected, and one of the samples are infected with the parasites, and the ornamental one are no single one fish are infected with the parasites. Okay, so we're going to the second result. The second result are talking about observation on the target organs. The type of group found to be infected with the parasite was E. fuscoguttatus, with infected target organs that 8 times more found in gills and 3 times more observed in the fins. And the results of this examination indicated that the prevalence of parasite that infected samples can occur based on the fish species, which shows that 100% of E. fuscoguttatus were infected. The result of this observation can be seen in the table in the left. We can see that there are some species that this research sampling. There are 4 species. The first are E. fuscoguttatus, and the second E. coiodes, and the third are C. altifelis, and the fourth was P. leopardus. And from all the species, only from E. fuscoguttatus that found the parasite that infected the fish, which are the age of the which 8% were found in the gills of Ephus coagulatus, and the three other parasites were found in the fin of Ephus coagulatus. And if we can see in the figure in the right, there are some sample, some documentation of the parasite that infected the fish, gills, and fin. Okay, we move to the third results. Please scroll down a little bit. Okay. Okay, from the infected group of fish species, which the Ephuscoguttatus, we examine these species to let us know the prevalence of parasites to determine which commodity are the most infected by the two species of parasites. The result of parasitic prevalence in table left showed that Trichodina sp with a prevalence of 10%, contributing the total prevalence, which is 11.25% of infecting parasites. While the Trichodina sp had infected 8 samples, while the prevalence of Diplectanum sp is only 1.25%, with only one infected sample. These results also show that all the parasitic infections occurred on the species of E. fuscogitatus, mostly coming from the fry commodity. Okay, maybe the discussion can present by Mr. Aviji.
Am I audible? Hello?
Yes, you are.
So the discussion was the occurrence of Trichodina was higher than the Plectanum species. Excessive mucus from infection caused the body of the fish to look radiant but the skin color turned dull. So the second one was fish attacked by Trichodina will also appear weak with some injuries to parts of the body, damage to fins and injuries in the gills. The infection by Trichodina is likely to cause damage to epithelial or epidermal cells and disease growth to death. The third one, the grouper who was attacked by Diplectanum species had a characteristic of partly wounded body and mouth. We call it hemorrhagic damage, plaque, caudal fins, swollen liver, and infected gills that look slim and pale. The Diplectanum sp will stick to the gills and disturb the breathing. However, it is also necessary to consider the tolerance of fish to glacial acetic acid or GGA administration. The use of natural ingredients can also be used as alternative in prevention of parasitic infections.
Hello, I'm audible. Hello.
Yes, correct.
Go
ahead.
Yes, thank you. Here a conclusion. Trichodenia and Diplocatinum were the species of the parasite identified in the grouper fish infection with Trichodenia was the dominant parasite. The total prevalence of parasites infection was 11.25% from the total of 80 grouper samples taken from fry. Consumption and ornamental fishes, future action can be planned to reduce the prevalence of parasitic infection by understanding the types of parasite and controlling the aquaculture condition to prevent infection. Acknowledgement. The acknowledgement of the prevalence. The presenter would like to thank the author for the knowledgeable studies that serve as a reference for this poster. We also acknowledgement acknowledge Institute Technology Sipulo Nopebar Indonesia for hosting Tarlac Agricultural University for hosting and the internship program, especially for the biology department, and terima kasih banyak. Here are reference.
Okay, thank you, Group 3, for the presentation. Now, Sir Edwin, you can go ahead ask the question to Group 3.
Yeah, here on the research design, you wrote about feasible on frying, consumption, and ornamental fish. What is the difference among that fry, consumption, and ornamental fish on the research design? Because as far as I know, grouper is only for what is the ornamental. That's why I'm curious. So explain me what is the grouper fish on these three categories.
Okay, Patwin, let me answer the question. So, in this research, they use three different kind of grouper fish. With the first one are the fry fish. The fry fish are I think it's kind of fermented or like maybe what we call ikan asin. Sometimes some kind like ikan asin, Pak Edwin.
Dried fish, you mean?
Yes, dried fish. And the second one, the consumption commodities are the
Fresh.
Yes, sir.
Yeah, consumption is for fresh.
Yes, the
Dried.
first fresh meat that we
Not fry, yeah,
consume.
dried, you mean?
Yes, yeah.
And ornamental?
And ornamental, we already looking for googling, and there are some kind of grouper fish that has unique color, and the fish can yes. I'm so sorry. I think I can show this.
Okay.
This one are the. I'm so sorry. Okay.
It's okay. No problem. No, it's clear now.
Yes.
And then
Right.
from the result,
Yes.
You wrote consumption. This is seed. What is seed on your result on the table? So, yeah, just go ahead on the table. Yeah, if you highlight that, it's seed, consumption, and ornamental. You mean dried? You mean is it like that? I don't understand seed.
Yeah. Yes. Yes. But when I think we also we didn't also know why the author typing seed in this table instead of dry or fry fish. I think there are yeah, something like typo maybe.
Yeah.
Yes, sir.
And then what is the difference between Fuscogatus coides? Do you know what is the basically different among these? E Fusigatus, E coides. Do you know what is this? Fusigatus. Seems that in Fusigatus there is a lot of infected organ. Why? But the other like Alcoides, Altifolis, Leopardus, no infected organs, zero. It's quite interesting. Right?
Okay, Pak Edwin.
Yeah.
I will try to answer maybe. So in this paper there are only two parasitic parasite that explained in this paper. Only I think. Wait a second. There are only Trichodina and SP and Yes, I think each parasite has preference for their host. So I think maybe there are some other infected parasite that can infect this Ephus cogitatus species, but in this area, in this Buleleng Bali, there are only these two of types of parasite that examine.
Examine. Okay.
Okay, and
Okay.
yes, sir.
Yeah. Last question. Then in the discussion, seems that the trichodina has the fatal injury to the fish. Is it like that? Fish attacked by trichodina. Why? Is it like that? So, and this is also interesting that most of trichodina like to stay or attack on the dried fish, while on the other hand, on the consumption and ornamental, it is also trichodina also zero.
Yes, but when I think that's the interesting part that the author didn't mention why the dried fish also had the infection
of trichodina.
Yes, of the trichodina. I think when the fish are dried, the trichodina also preserve with the fish when they dry it. When the diet mechanism are yeah, I think something like that.
Okay, thank you, Dinar. It's enough, I think, for question to them. Thank you, the group 3.
Okay. Thank you, Group 3, for the presentation. Now we will have a break about an hour, and we will be back at 12:30 in Indonesia time and at 1:30 in...
Dinar, maybe I have some idea. We just pass it until the 4th group and then we have a break.
Oh, okay, sorry.
Make it quicker. Is it
Okay.
like possible? Because I'm
Yeah.
not really in well condition, so after
okay.
this I'm going to the doctor. So
Yeah,
if
okay.
possible, then we can take a break at 12. Then at 1 o'clock I can go to the
Okay,
physician.
yeah. Okay, sorry. Now Group 4. We will continue again. Maybe now you guys, Group 4, can start the presentation.
Thank you, Miss Dinar and Pak Edwin. I'll let my teammates to start the share screen. Am I audible?
Yes, Gege, you are.
Okay, thank you. So good afternoon, everyone. It is pleased to join you as the last sessions of this program. I am Gege or Geoloverenisasi from the group 4. And we are here today under the theme of biofertilizer. We have want to deliver our poster presentation with the title of the production of plant growth promoting bacteria biofertilizer from organic waste material and application on the growth of corn or zea maize. And this poster has been done by our group, Ella C. Fernandez and Melchor D. Gragasin from the Department of Biology in Tarlac Agricultural University, and me myself, Gelo Ferenesasi and Muhammad Habib Dwi Fernando Brianto, from the Department of Biology, 10 November Institute of Technology Indonesia. For the introduction, I will give the opportunity to my teammate Melchor.
Thank you. First, the chemical fertilizer. Fertilizers play an important role in agricultural system, mainly in increasing yield. Synthetic fertilizer were a part of the technology that contribute to the green revolution, and millions of tons of chemical fertilizers have continued to be used in the world over. However, excessive application of chemical fertilizers has resulted in adverse environmental impacts and ecological impacts, include water body pollution and algal bloom, land degradation, food and food products contamination, adverse human health effects, greenhouse effect or ozone layer depletion, and general imbalance of the ecosystem. These adverse effects have led to the introduction of microbial inoculant biofertilizers for plant growth and development, which include plant growth, promoting microorganisms such as nitrogen fixing and phosphate solubilizing microorganisms. Second figure, nitrogen fixing and phosphate solubilizing microorganisms. So the solution is used by fertilizer to promote the growth of plants. Biofertilizers is any substance which contains living microorganisms that can inhabit the rhizosphere or soil and promote the growth of plant by promoting promote I'm sorry, promoting the availability of the primary plant nutrient when applied to the soil. Purpose of plant or seed. On the other hand, describe biofertilizers as material that include manure and plant extracts which contain microorganisms that promote plant growth. This group of microorganisms is known as plant growth promoting bacteria or PGPB, some of which promote plant growth by fixing atmospheric nitrogen and solubilizing phosphorus. This application is based on the use of decomposing organic matters, crop remains, animal sewage, and microorganisms to enhance nutrient in the plant rhizosphere. Soil stability, quick decomposition of organic matter, and mycorrhizal symbiosis association. For the purpose will be continued by Miss Ellen.
So the purpose to compare the performance rates of the biofertilizer to those of the nitrogen, phosphorus, kelp, NPK, chemical fertilizer, and the control of corn grow. So the materials and method, culturing of microorganisms, production of liquid biofertilizer, isolation of microorganism, characterization of the experimental soil, biofertilizer and chemicals, synthetic fertilizer, and application for plant. So the materials used are fish, soil, sample from the soil, repair taken by 5 cm beneath the soil surface, and we need organic waste like dry grass, water hyacinth, so leguminous plants, rice craft, wet craft, and the moringa leaves, orange, banana, grapes, cucumber, poultry titers, sewage, and brown sugar. Then there is also tools used, including microgeldal flash, weighing balance, breaker, retort stand, and the bottom flash is a column of conical flash.
Okay, hello. Am I audible?
Okay, go ahead.
Yes, you are.
Okay, thank you for the confirmation, and thank you, Ella, for presenting the materials part. So for the methods that use at we have a lot of methods that conducted during in this paper, in this research by the authors, especially at the different stage of the production and the product testing. It is including culturing of the microorganisms, the production of the liquid biofertilizer, the isolation of the microorganism, and then characterization of the experimental soil, the biofertilizer and chemical synthetic fertilizer, and the application of the fertilizer. From the culturing of the microorganism, because the target of the research is to compare the PGGB bacteria. The authors use the Picosphaia agar medium. It is right now, currently, the medium is one of the most used medium for the PGB's bacteria, and the microorganism was cultured using 500 grams of the fresh soil sample from the soil reservoir collected 5 centimeters beneath the soil surface, and then also use 500 grams of wheat chaff and rice chaff mixed with water until a homogeneous mixture was obtained. The mixture then transferred to a covered container with depression in the center to allow air circulation and ventilation. And then the container was kept under shade and allowed to stand for 7 days. Next to the second part of the methods is production of liquid biofertilizer. About 9 kilograms of mixed waste, fruit waste, and moringa leaves were cut into tiny pieces and loaded into a container. It is mixed with 1 kilogram of brown sugar, 1 kilogram of the microorganism culture, with the ratio of 1:1. Then we mix it. The material were mixed properly together and until the mixture become consistent. Sterile water was added to the container and stirred in one direction for only a few minutes, and then the container was tightly sealed and kept in the dark place for 2 weeks without any disturbance. And then the mixture was sieved, and the liquid biofertilizer was transferred into a gallon and then kept in the shady, cool, dry place and ready for the next step, that is the isolation of the microorganism. The liquid biofertilizer that has been produced was cultured using two selective media, for azotobacter and phosphorus solubilizing bacteria respectively. I already mentioned that we use the Phycosphaia agar medium with the main components are glucose, Ca3, and NaCl. So in Phycosphaia, the sugar that used is glucose. The medium was diluted in 250 milliliters of distilled water and then sterilized in the autoclave in the temperature of 121 degrees Celsius for 15 minutes. And then it was allowed to solidify at the room temperature. The next is the isolation of phosphorus solubilizing bacteria. And then we characterize the experimental soil, the biofertilizer, and the chemical or synthetic fertilizer. And then we come to the application of the fertilizer, and then we can compare it to the chemical fertilizer. So the main distributed chemical fertilizer that contain nitrogen, phosphorus, and K is using the ratio of 15 by 15 by 15. Meanwhile, in the biofertilizer that was produced in this research, using the NP and K with the ratio of 2.5,
Okay, please,
3.5,
I cannot
and
see
2.5.
your result in discussion because it seems like freeze. Because you told about the composition and then there is no composition here.
Sorry, sir. We are still in the
Yeah.
methods part. So it is the last part of the application. So before the result and the discussion, in the application, sir, we because we compare the chemical and the biofertilizer, it is important that we highlight the difference of the ratio.
Next time you put the ratio because here there is no ratio. So that's when you quote ratio, I'm confusing.
Okay, sir. Thank you, sir, for the suggestion. I think that's all for my part. Please, next to the result and discussion will be further discussed by my teammates.
Okay, thank you for Q. And I want to explain the result and discussion for this research. So, for this research, in the first table, there was comparative analysis of the chemical composition of the soil, synthetic or chemical fertilizer, and then biofertilizer that showed that the chemical fertilizer has equal ratio of nitrogen, phosphorus, and potassium much higher than the ratio of the chemical the chemical chemical composition of the biology fertilizer, and that of the soil. So this is the different. The chemical fertilizers is more high, but the nitrogen, phosphorus, and potassium. And then in this in this table, the effect of the application of chemical fertilizer and biofertilizer on corn plant growth, that was see the biofertilizer consistently and significantly increase the height of the corn plant. It's compared to other treatment. At the 6 weeks, we can see the height of the corn treated with biofertilizer was observed to significantly increase to 100 9 190 93 centimeter compared to NPK and the control. Also at the 8th week, after planting, the height of the corn was observed to significantly increase to 225 compared to the other treatments. And as the indicated in this table, main heights of the corn plants treated with chemical fertilizer and biofertilizer, the highest value of standard deviation of the biofertilizer showed the improved effect of this treatment on the growth performance of the plants over the other treatments. This very high. There was 13.7, and then uh, for the production of corn, given the biofertilizer has a higher number two than the other treatment, that was 29, and the chemical fertilizer is just 18, and the control just 12. In the main weight of corn, that given biofertilizer is also greater weight than the other treatments. And and this is the graphic representation of mean value of the growth of corn plant that planted with the two fertilizer and the control. So uh, with the in this graphic, the blue color is the control, and the red is soil with a chemical fertilizer, that was NPK, and the green color is soil with biofertilizer with the microorganism. And this is a figure above represent corn plants, for A, its control plants, and B, its corn plant treated with chemical fertilizer, and the C is corn plant treated with biofertilizer. It could be noticed that while figure B and C demonstrated a finding of increased growth against the control plant, apart from increased growth, also demonstrated protection against insect or pest attack as shown by the healthier leaves. You can see this not healthy, and BNC is more healthy leaves for the plants, corn plants. So for the presence of microbes and then the trace elements in the biofertilizer might have contributed to improve performance of the corn plants treated with a biofertilizer as against the performance of the crops treated chemical fertilizer and the control without microbial inoculum. Maybe for the next, my friends can explain.
No?
Oh, okay. For maybe for the conclusion, for this research, based on the findings and the observation made, it was concluded that the biofertilizer a higher crop performance efficiency than the chemical fertilizer. The plant growth promoting bacteria or PGPB isolated and used as biofertilizer in this research work also demonstrated the ability to protect the corn plants from pathogen or insect attack, and therefore can serve about biofertilizer and biopesticide or biocontrol again.
So that concludes our presentation. We would like to acknowledge our teammates, especially me and Habib from ITS. What would like to acknowledge our friends from the Tarlac Agricultural University, Malcor and Ella. Thank you so much. And for all the committees and the lecturers, we will also would like to say thank you for the opportunity that has been given during this internship program. We hope for the better collaboration and we can see meet uh, on offline, either in Surabaya or in Philippines. Thank you so much, and that concludes all of our presentation. Back to Mbak Dinar.
Okay, thank you, Group 4, for the presentation. Now, also Edwin, you may ask the questions for Group 4.
So number 1, what kind of bacteria inside the culturing microorganism? Because PGPR, plant growth promoting bacteria, is really famous now. Do you know what is the how you call it, like what kind of species inside the bacteria? Is it like special bacteria or something like that? Have you already searched that?
Yes, may I
Mhm.
answer?
Mhm.
Okay, for the uh, journal that we use, the wait, uh,
Maybe bacillus or something like that. Have you checked it? Not yet. No, understand.
yes, for a number of bacteria that we use and in the plant growth promoting biofertilizer, it's include Bacillus species, Pseudomonas, and the
Okay.
Azotobacter species.
So it looks like specific for the promoting bacteria. Second one, what kind of organic waste used here? Because in the material method, we wrote organic waste. Is it like junk or something like that from the rest of the plant or rest of the fruit or organic waste? What is inside in the journal?
Hello, sir, Mr. Edwin. Am I audible?
Yeah.
Yeah. So the materials that use the organic waste that I mentioned before is most of the time is mostly is fruit waste like banana peel, orange peel, uh,
Why? Why? Because a lot of also organic waste from leaves or roots or bark or wood. Have you any explanation?
so
Why?
so basically the main purpose of the research was to eliminate a chemical fertilizer. So a lot of this fruit waste that has been used as the material for the biofertilizer already contain the natrium and phosphorus. Already contain
Nitrogen, yeah.
nitrogen and phosphorus. So
Okay.
that's why I already mentioned before that the phosphorus nitrogen that added to the biofertilizer has little has little it is uh, so much a smaller ratio rather than the chemical fertilizer that has been used right now. So that's why there are only certain organic waste that
Yeah.
used as the organic material, sir.
Okay, good. The last question is about, seems that this is really, the result was striking, yeah. I was terrified because, I mean, in comparison with chemical fertilizer, it gives or it yields much better result. As far as our opinion, when you do the chemical fertilizer, it should be more because it's pure. But in biofertilizer, the result give more. Is it any clue on this? Why this is really significant?
Sorry, sir. I'm still audible.
Yeah.
Yeah, hello. So the because this is a biofertilizer, I think from the our group discussion, our analysis was because it has enhancement on the PGPB organism. So I think that's why the result is verified and pretty much enhance the producing rate because it's already the biofertilizer has been produced. The composition of biofertilizer is creating the symbiotic interaction with the bacteria also. So I think that is the main reason why the biofertilizer significantly has better producing rates. Perhaps my and the other teammates has their own opinion.
Any question? No? Okay, I think it's enough, yeah. Dinar.
Okay. Thank you, Group 4, for the presentation. And now we will have time for break. Okay, we will have a break about an hour, and we will be back at 1 p.m. in Indonesia time and at 2 p.m. In Philippine times.
Okay.
Okay.
Or is it okay if we make it quicker or not? It's better, up to you.
Excuse me, sir?
Because now it's 11:48. Shall we start 12:30 or 12:45, I think.
Oh, okay. We can start at 12.
Yeah, earlier it's better, I think.
Yeah, okay. Then we will start again at
12:45 or
yeah.
Philippine time is 13:45, I think.
Yeah.
It's enough, 45 minutes.
Okay.
For eating.
Okay.
Okay. Okay, see you in 1 hour, yeah?
Mm-hmm.
Shall I leave then? Okay.
Thank you, sir.